Okay, there we go. All right, so I've got PCR, ability to copy one little tiny segment of DNA into billions of copies, for which I can figure out what in the hell it does. I have polymerase chain reaction, the ability to see the genetic information in a visual representation. And by visual representation, by weight and charge. If I have a known segment of DNA and compare that to the DNA in question, by just having the similarities between the two, you can say, okay, this is related to this individual. There's some kind of genetic link here. We don't know what it is. You can do more research into it. We've been able to clone specific segments of our genome. We've been able to insert that gene that we know what it does, like produce insulin, produce a protein of some sort. And we've been able to put that in a bacteria and harvest it from a bacteria. We are able to do that in eukaryotic organisms as well. And that leads us into this concept of genetic engineering, and it's the ghost in the room that I've kind of been talking about. All right. Being able to take a favorable piece of information, of genetic information from one creature, and insert foreign DNA into another creature. And this is what genetic engineering is. As soon as we take that genetic engineering and insert it into another creature, now we have a GMO. And again, this has been going on since the 70s with crops, okay? With putting sequences in the genome to, let's say, corn to make it resistant to certain herbicides, okay? We've been doing this for a really long time. They do a ton of research with corn. We know it's DNA. We manipulated the bejesus out of it. And so we do a ton of research. That's one of the biofuels now. That's why it's one of the biofuels now, because we've been able to really manipulate the hell out of that thing and get it to produce stuff to where it's really going to benefit us a hell of a lot more. As soon as you start introducing DNA into other different creatures, and we've done it a ton, they are referred to as transgenic. Now, this can be utilized, again, for the goods of understanding and knowledge, so academic. This could be done for medical reasons, making insulin, like that picture was back there. Again, agricultural use, making it more easy to grow crops at all times of the years, which has also made, fueled maybe obesity issues as well, but whatever. And some industrial purposes, making detergents, making industrial cleaners, okay? So we have a lot of great things that have come out of this concept of genetic engineering. But for me, I always look at the evil. I look at the bad in people. I don't know why. I don't know what that says about me. But I like to look for the good in people. I really do. I love to give people the benefit of the doubt. Okay? I love to be positive. But I've lived a very interesting life. So I know most people are bull. And I don't trust people as far as I can throw them. I trust my family, and that's really about it. And I don't know what the hell that says about me, but good God, I sound like an awful person. I'm really not. I just don't trust anybody. I've been duped before. So, you live a jaded life. You get jaded. So I look at all of the evil aspect of it. Again, I get it. If my kid had a genetic disease and I could do genetic engineering to cut that crap out of him so he doesn't have that affliction anymore, or to insert some DNA into him, do some stem cell treatment to where it can heal and repair and he doesn't have that issue anymore, you're damn right I would. Okay. But what's the line? What's the line where we stop? Right? where do we stop? Because you know it's not going to stop at just genetic engineering for getting rid of certain diseases, which sounds fabulous, right? Have you ever watched? I love random tangents here. Altered Carbon. It's a show on, I think, Amazon Prime. I think it's a series. If you like sci-fi and it kind of talks about this stuff without talking about it, it's fascinating. Uploading the human conscious and what really is Netflix. It's on Netflix. Okay, and what defines being human? To me, that's what all of this kind of dovetails into. Because I mean, if you're going to grow a clone to harvest body parts out of in case you screw them up, I mean, who's to say that clone can't be something? I don't know. This is a fun discussion when we're all face to face in class, because then I can start yelling at you and getting you to think, connecting dots, conspiracy theory nonsense. This is good fun right here. This is good fun. There's a bunch of genetic diagnosis. Has anybody done 23andMe or any of the genetic sequencing out there? If you have, there's various different interesting. You can do a where did I come from type of genetic diagnosis. You can do one for medical screening as well. It's pretty interesting. I see one individual here has done it. It's pretty fascinating, some of the information we know. Um, it doesn't tell you if you're going to have, if you have the genetic sequence that codes for certain things. It doesn't say that you're going to have it. It talks about it in vague terminology, but it talks about the potential. My mom, she passed away, and well, both my mom and dad are gone. But my mom, she had ovarian cancer, and she always wanted us to do genetic profiling to kind of see if we had that gene and to figure out what we needed to do and stuff like that. So it's pretty interesting, and I did it for grins and giggles a few years after she passed. And I don't have that BRCA gene, the BRCA gene that increases the likelihood for those type of cancers. So not that I'm a woman and have ovaries either, but breast cancer can be something that a man can get. And I don't know if the BRCA gene does anything with prostate at all off the top of my head, but that would be my bag, and because I'm a guy, right? Okay, so yeah. Anyway, but you can do this to kind of figure out all these various different. There's a ton that's on there. If I pull it up next time, I'll show you some of my results because it's pretty fascinating. Some of the stuff. Did you find anything in yours? Let's see here. David, did you find anything in yours that was interesting? If you want to share, you don't have to share, but I thought I'd ask. Gene therapy is this genetic engineering technique that we use now on non-mutated genes. That's a clickable link that you can go to if you want to see all the approved gene therapy products that are out there. It's not a very exhaustive list. It's actually, I was shocked to see as few clinical trials as there are. I know there were a few more at some point, so those are obviously done and they're doing their analysis of them, so they weren't listed on there as something currently ongoing. But these are some of the approved products here and the clinical trials that are ongoing. So if you are curious about it, there's a clickable link that you can go and kind of see kind of what they're doing. A lot of, they're all within the same realm of stuff, but it's all pretty interesting here. And this is a great picture that I got off of the the CDC's website, I believe. Um, I thought I had a spot there where I got it from, but this kind of shows using this vector, this virus as the vector here, and how we've inserted our DNA into that virus vector. And then you can see we utilize viruses quite a bit as well as a vector. Viruses get in and very easy into host cells, into our cells. And as you can kind of see, they can squirt their genetic information into the genome, into the nucleus that houses our genetic material, obviously. And you can get these cells to basically do what the virus tells them to. So this is kind of a much more better updated kind of visual of that concept of introducing a plasmid.
Genes make us who we are. Are we tall or short? Do we have blue eyes or brown? Our genes hold the answer. What if part of a gene is defective or missing? Or if a gene changes or mutates? It can cause a disease like cancer. Enter gene therapy. Among other things, scientists can replace a bad gene with one that is healthy, add a new gene to perform the function of a defective or missing gene, or they can turn off a problem gene. How do they do it? Say scientists want to add back a healthy copy of a gene to cells. They need an efficient way to do it. So scientists use vectors or vehicles to deliver a gene. The healthy gene hitches a ride in the vector. Viruses are one kind of vector. Scientists modify a virus to remove its ability to cause a disease. The modified virus can then be used to carry the healthy gene into a cell. Gene therapy can happen on cells inside the body, or can happen on cells outside, and then transplanted back in again. Genes make us who we are, and gene therapy holds great promise for a healthy future.
And that's the FDA. They are painting it with a very lovely picture. Obviously, for me, I think there's more pitfalls to it, as I've stated, but the FDA paints a very favorable picture of that. Here are some applications for this, because like I said, this has been going on for a very long time, longer than all of us have been alive, even me. So here are some kind of applications to this, all right? Because there are some applications. Again, I may have a handful of short answer questions. I'm sorry, multiple choice questions based on some of these things. So that is where you would potentially see them. Okay. Vaccines. Vaccines are what you get throughout the course of your early life, and you're supposed to do it into adulthood as well. Most adults don't. But there is a vaccine sequence for adults. But with a vaccine, it is basically something that's been manipulated, a virus. A lot of the diseases that we are getting there to prevent are viral in nature, okay? Viral causes in nature. So by manipulating the virus to not be so evil to us, we take out all the information that causes all the harm, but just leave the information on the outside of the virus that says, hi, I am Mr. Virus. Remember me. This stimulates the immune system to recognize that viral invader, kill it, mount an immune response against it, and then create memory to where if you are ever come into contact with that virus again, your body remembers, mounts an immune response, and kills it off, and you're perfectly fine. we have done this whole genetic engineering with these different viruses with vaccines for a number of years, have made a number of different vaccines in this regard. And one of the more recent ones is H1N1. And I don't know how effective flu vaccines really are. I don't believe they're that effective at all, to be honest. But they have the potential, if they are right in predicting the specific flu strain that does come over here to America, to this part of the world from Asia. And if they're wrong, well, it's not an end game result, but you're still going to get the flu potentially. But we've done a lot of this vaccines with flu, different vaccines. We've done a lot with antibiotics. To me, this is both the gift and the curse. We're able to grow a huge amount of antibiotics in these basically big tanks with various different bacteria or various different fungus. Kind of like if you've ever gone on a tour there of Budweiser or here in town Boulevard, how they make beer. That's basically what they kind of grow all these bacteria and fungus in. And they just harvest it. It's a very basic rudimentary thing called gene farming, basically. But they've done it, as you can see that for penicillin, on a non-antibiotic aspect of it. They've done it with fungus to make fish oils and whatnot. So there is a source of fish oil, omega-369, that comes from grown in a fungal source. So pretty interesting in that regard. So this is something that this genetic engineering is something that happens on a much more often scale than what we really think of. HGH, human growth hormone. This is what they use on children right now who have disorders associated with dwarfism and whatnot, various different types of growth disorders, not just dwarfism, to get them to stimulate the stem cells basically within the growth plate to divide and grow so that their bones can get longer. So it's pretty interesting there. You can see insulin kind of gets talked about quite a bit. You can see that we used to use pig insulin, but there was a ton of allergic reactions associated with it. So that's why they started doing the recombinant bacterial DNA issues and growing it in vats. Here's gene farming, guys. I keep mentioning it. What the hell is it? Here it is. Okay. Again, I've taken the specific thing that I want. This is the gene that codes for the human growth hormone. This is the gene that codes for thyroid hormone, whatever the hormone is. Okay. Whatever the thing that I want to be made. Okay. I take that genetic sequence out of me, out of my gene. Okay. I insert it into a vector and get that to get inserted into another different animal. Goat, cow, whatever. That genome, that foreign DNA, okay, can now be taken up into my new goat, let's say. And I can then milk that goat. I've inserted this genome to make human growth hormone, and I've injected it into a specific gene that I know is where milk production occurs in the goat. Goat gets milked. Milk has the human growth hormone in it. Ta-da. There is my gene farming. I can insert this transgenic piece of genetic information into a goat, and I can get it to reproduce. And I can produce a bunch of clone transgenic goats. All of them can produce it. And I don't have to farm or splice this into every single goat at my farm. I can just reproductive clone these suckers. This is going to be more for, obviously not a farmer. It's going to be more geared towards pharmaceutical companies because this is going to cost billions and billions of dollars to do. Where we do it is mostly with plants getting desirable traits. Okay. Pest resistance, better nutritional value, better shelf life, better transportation time, longer to ripe as it's plucked off of the plant. All of these things are possible because of the manipulation to the DNA of these various different vegetations. The problem, the one potential problem that I see with this is that you've manipulated these genes so much. What's the true source? What's the true genetic information that is within that plant now? It's called wild type. The wild type DNA of plants out there that have not been manipulated, they have everything inside of them from the very beginning, passed down from one generation to the next. They have the entire genetic sequence within them that's able to potentially adapt and survive. So once we've taken the genetic sequence of a plant and manipulated it to kingdom come, it's not the wild type anymore. It's man-made. Who's to say something can't be introduced in the environment that this plant just can't overcome? It doesn't have the genetic information anymore. So now you can see the potential downfall for this huge amount of manipulation that can occur and that has occurred since the 70s when it comes to farming, vegetation. It seems super easy to us talking about it here that we can keep. Here's my transgenic crop. It is right here on this plot of land. And here it sits. I got a field. It's open here. I got a field open on all sides of it. Okay. So that little pollen can't go from this crop here and get next door to that crop of corn. But what lands on these things? Bugs, flies, bees, grasshoppers. They get all that sticky pollen and whatnot all over themselves. And then they just fly off and go someplace else. So can this potential genetic information be easily moved around? Yes. By cross-pollinating a wild type with something that's been genetically engineered, what are we going to get? We don't know. What does that do for the potential harvest? You don't know because you didn't grow it in a lab. You haven't grown it in a crop field. That's a laboratory field to figure out what the hell it does. Is it fit for human consumption? Well, first we got to feed it to, this lab animal and see if it doesn't croak over and die. And then we can start introducing it to bigger and bigger, more complex organisms until, okay, it's deemed safe for adults. These are the potential pitfalls that come with transgenic plants. Transgenic animals as well, but transgenic plants. Being able to grow corn in places that it wasn't able to, fantastic. Being able to grow potatoes in places where you couldn't do it before, fantastic. this solves a lot of hunger issues. We don't have a lot of hunger issues here in the state with, it's more access to food. Okay? There's plenty out there because there's plenty of it thrown away every year, every day. So there's plenty of food out there, but in places around the world where starvation is very common, this makes it very easy to introduce crops to a region so we don't have little children dying from starvation. PCR, gel electrophoresis, molecular cloning, reproductive cloning, recombinant DNA, transgenic animals, transgenic plants. It all leads to understanding the genomes for all of the various different creatures and animals out there. Genomics is this study. This is what the Human Genome Project did. Our understanding of this genome through all of this knowledge of understanding how to manipulate it, how to figure out what the hell it was to begin with, and our ability to sequence, because it's billions of segments long for some of them, being able to get all that information out in a fast amount of time is something that supercomputers and scientific advancements has allowed us to truly understand the genomics of every single animal out there and at least sequence them all, not fully understand them all, but at least sequence pretty much all of them out there. We have a few different types of genomics. They make a great matching question on an exam. When it comes to this concept of understanding the genome and mapping it out, all it means is just we list the genes, where they're at, and what they do. Okay. And I have some links, I believe, here in a second to the Human Genome Project, where you can get an actual list of all the genes in the human genome, because those are the ones you honestly really care about. You don't care about a hamster, I imagine. And this is what 23andMe and some of the other ones out there have done by looking at the specific sequence, the genomic map, and taking our DNA and comparing it against it. You can see if you have a variation in that specific gene location. And if you have a variation, that gives you the potential for some of these really negative things that 23andMe looks for.
In 1989, a group of scientists was assembled to decode the human genome under the direction of Dr. James Watson.
One of Watson's big motivations here was to try to recruit the best and brightest from many different disciplines, biology, but also physics, chemistry, engineering, because all of those would have to get together if this was going to work.
Many of the biologists, and I love them dearly, of course, were dragged into it kicking or screaming in the beginning because they failed to see the tremendous value that such an enterprise would provide the field.
The talent turned out to be all over the world, so you have to basically make it possible for those bright minds to get engaged where they are. They're not going to all move to join this effort.
Genomics has fundamentally changed the way we do science. It brought the notion of team science and collaboration to biology.
Curious thing about collaboration, it's also intensely competitive. Each person wants to achieve something personal. You know, they want to make their hit, you know, on the problem, but then immediately they want to share it with the other, and the other learns immediately. And so you ratchet each other up.
From the beginning of the Genome Project, there was a fairly strong sense of enthusiasm in the public sector, the private, and academia.
We had lots of support from Republicans, partly because Newt Gingrich was interested in science, and I knew it. There's no partisan difference in everybody wanting to live as long as they can.
I think there's a lesson there. It isn't a big monolithic project. It was a big challenge, but it brought people together. And I think that's what we have to do in the future.
When the Human Genome Project was started, it's important to note the technology of sequencing was not developed yet. So it's, in a sense, a project that was launched with a wing and a prayer.
We couldn't sequence it at that time. The machines were nothing like fast enough. There were no machines even. It was sequencing was done by hand.
I thought we might have to do it brute force, and I was quietly prepared to use the old-fashioned technology just because it was so important to get it done.
There are a lot of things, you know, that would make life easier that just hadn't been invented yet.
It'd be a 100-year project, like monks working to copy a book at best.
Bit by bit, we got into more and more large-scale sequencing. But the sequencing is enormously important because it provides the possibility of searching by computer.
When I showed up at Washington University one day with a computer terminal under my arm, people looked at it and said, "What are you going to do with that?" It's a genetics department.
Genomes were supposed to be too complex to be able to reassemble them in the computer.
Without computers, we simply would be dead in the water.
Computers and genomics were made for each other. Genetic information is digital. It's like binary code. It just happens to use 4 digits instead of 2. It is amazingly accurate. And that's what biology gets from digital information.
The Human Genome Project contributed a cultural change about data sharing because the Genome Project was about sharing data almost immediately. And while it seems obvious now, it wasn't obvious then. That required significant effort.
I think I'm quite lucky as a scientist because I grew up always sharing data. The community around me, the genomics community, the worm and fly communities as well. It's just a default that you're going to share everything. You're going to share the code. You're going to share everything by publishing, but you're going to share it before publishing. You're going to share half-cooked ideas. And in fact, this is a wonderful thing. It's liberating.
It would have been virtually criminal to try to hoard discoveries that meant the difference in life and death in people.
This is information which we'll be using for centuries. The sooner we get it and everybody has access to it who wants it, the faster we can move in terms of understanding how life works and how disease occurs.
Patients invest a certain degree of fear about genetics. Who's going to find this out? How will it be used against me?
A lot of the ethical and social considerations that we're struggling with today really have historical roots in the early 1900s in the United States, where we had many states that had laws that allowed us to forcibly sterilize people and prevent them from having children based on whether we as a society felt that they were genetically fit.
So at the beginning of the Human Genome Project, we got it right of bringing in the social scientists, the ethicists, the lawyers, and we now have helped to provide a whole generation of scientists that are focusing on the ethical and social issues of genomics.
The sequencing of the human genome was a monumental achievement. Decades of work, thousands of scientists, billions of dollars, and we get this first roadmap for what makes up the human genome. It's an incredibly exciting moment. At the same time, it's just the beginning of what's really a much longer voyage.
And let's again be audacious, but let's not be unrealistic about what's possible.
I think the next 10 years are going to see some exciting developments in genomics, particularly those where we start to use genomic information as part of routine medical care. And that'll be applied to dealing with diseases like cancer, hypertension, diabetes, and Alzheimer's disease.
I dream of the day when every young mother brings a young girl or a young boy home from the hospital with a little gene card, and they say, this is the way you should prepare this child's health care. It's exhilarating.
I'll click on those in a second. But that's kind of where they're going with pharmaceuticals, or trying to go with pharmaceuticals. They're trying to get to that, what does your gene say, and apply medication specifically according to how your body can handle it. So that's where, they're kind of coming from. When it comes to applying genetics, guys, I mean, this is not earth-shattering information here. Why else do it? Why else do it? Okay. If you can get a sequence of the genetic information and prevent a lot of this stuff, holy smokes, you could potentially save a lot in terms of what it costs the American people. And I'm just using America as the standard. Okay. In terms of healthcare cost, you could save billions of dollars, billions and billions of dollars. So why not? Why not do it? Here are the most common types of genomics. Okay. This is what I just talked about, pharmacology genomics. This is basing your drug use, your medicinal drugs, based upon your genome. This is kind of the new one here in terms of it, in terms of its potential application. This is the one that they've been trying to push a hell of a lot more as of late. Metagenomics, guys. This is understanding how species grow. When it comes to this one, why is it important? This is more of kind of understanding evolutionary mechanisms. When it comes to metagenomics, understanding the genome that a specific species has and how it's able to interact with its environment. If it's able to interact with its environment really well, that's called better fitness. And if they have better fitness, then they're able to find a partner and have sex with, and then have offspring that carries their specific genetic information into that next gen, so that they have the advantageous traits within them to be just as fit for that environment. So metagenomics is kind of this subset of genomics that's broken off in terms of applying evolutionary patterns to figure out who has the fitness and what does that fitness mean for this environment, and what other individuals, what other species have similar genomics to survive similar types of environments. So it's pretty interesting there. Mitochondrial genomics. Mitochondria contain their own DNA, and it doesn't change a whole hell of a lot. It's passed from mother during the process. So they do a lot of mitochondrial genomic work to trace genealogy. So that's the most common one that's utilized in that aspect. And then this last one here, proteins dictate function. Proteins get coded by the DNA, which is transcribed into messenger RNA and then translated by tRNA out in the cytoplasm. So protogenomics is the study of proteosomes, the specific segment of the DNA that codes for a specific protein. Some of these are turned on and off by various different types of environments, different types of diets. So proteomics is a pretty useful one to try to figure out, what genes are, what can get them to be expressed, turned on. So when it comes to kind of understanding disease, proteomics is one of the subsets that is utilized of genomics for that application. There's also another subset, this one called nutrigenomics, where it looks at how nutrition affects the gene sequence, which affects protein expression, which is proteomics. I want to come back here. I want to click on this. Yeah. Where is my... So here is this kind of human genome map. And here you can see it's all, and this is just a link to it. You can click on it. There's clickable links in the other one there to kind of get in there and look at all the individual ones. If I just go in there and get really close here. I mean, here is chromosome number 1, and you can see, oh my God, you're like, holy smokes, what does it code for? Cataracts, homocystinuria, neuroblastomas. This is rabiomyosarcoma, alveolar, neuroblastomas, glaucoma, Ehlers-Danlos syndrome, connective tissue thing, breast cancer, ductal breast cancer, hyperprolipidemia, prostate cancer. I mean, look at all this. Look at all this. Colon cancer, hyperlipidemia, familial. I mean, this is so cool, right? Ewing sarcoma. Oh my gosh. They have the X and the Y chromosome over here. You can see there's a hell of a lot of stuff on that X chromosome. Not a whole heck of a lot of stuff on the Y chromosome. Really, the big thing here is the sex determining factor, the sex determining region there. That determines whether or not you're going to be a dude, male. Gets those proto reproductive organs to start becoming male. So pretty interesting there. Interleukin-3 receptor, that's kind of an interesting one. Interleukin is kind of like some growth hormones type stuff. But I mean, there's a whole lot of stuff here. I don't know what half this stuff is. These are just some weird, crazy different things. But you can see there's a lot, prostate cancer's over here was also on number 1. So there's a lot of information here, and just truly understanding how all of these genes interact with one another, man. It's just, this is so fascinating that we have all this information. And again, just a variation right here in this little bitty sequence could be the difference between you having something and not having something. And you can see how supercomputers really help to, because I think when the Human Genome Project first started, like the one dude said, it was going to take like 100 years. They were talking about, something like in 2050, finally having the human genome sequenced. As soon as we were able to have much more better computers there, I mean, they got done in like 10 years or some nonsense. It was really fast. So very fascinating, very kind of cool information here. And if you click on that side here, there's all the individual ones, and you can click on them, chromosome viewer. You can kind of see what they look like. Okay, and you can click on the individual ones, and then you can see kind of all their stuff. It has a nice little viewfinder on it as well. So it's pretty interesting. But there's a lot of information there. There's research, publications on it. So, I mean, there's a bunch of cool information on here, guys. If you're curious, I hope you are. Go in there and click around and look around and get some information there. I know we went a little long today, but I want to get this thing done because I want you guys to take this damn test and be done so we can continue to move on. I've kind of taken a little bit longer on this one here, okay? Wednesday, name over there in the box if you like credit for being here, okay? Wednesday, hopefully the world will still be in one piece and we will have class. We will have a class over the